Hello, everyone, and welcome to today's webinar, PCR Testing Reveals that SUC is not so golden of a standard of care. I'm Antonina Salcido of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, please visit thermofisher.com. Now let's get started. I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you would like at any time during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. I now present today's speakers, Dr. Becky Winslow, Clinical Science Liaison at Acutus, and Dr. Mayil Shanmugam, Senior Scientist and Director of Molecular and Microbiology. For a complete biography on our speakers, please visit the Biography tab at the top right of your screen. Dr. Winslow, you may now begin your presentation.
Hi, my name is Dr. Becky Winslow, and I and Dr. Mayil will be discussing with you today polymerase chain reaction testing and how it reveals the standard urine culture is not so golden of a standard of care. So let's jump right into the impacts of urinary tract infection, as they are quite extensive. Urinary tract infection affects approximately 150 million people worldwide. Urinary tract infection is also the most common outpatient bacterial infection in the U.S. Urinary tract infection leads to 2 to 3 million emergency department visits per year. It leads to approximately 10.5 million office visits per year. It causes 3.5 billion in healthcare-related costs annually. And last but certainly not least, urinary tract infection leads to workforce presenteeism and absenteeism. So let's look at the standard urine culture and some of the technical challenges associated with the standard urine culture. True or false? The standard urine culture was validated for use in identifying lower urinary tract microorganisms. What do you think? False. The standard urine culture was not actually validated for use in identifying lower urinary tract microorganisms. Yet, for more than 60 years, the medical community has relied upon the standard urine culture for identifying uropathogens. The standard urine culture was never validated for use in identifying lower urinary tract infections like bladder or urethral infections. And those, cystitis in particular, represent the majority of urinary tract infections. So let's look at why the gold standard for identifying uropathogens is not so golden after all. The standard urine culture results in a high rate of false negatives. Why? Because the standard urine culture is biased to grow E. coli, and with standard culturing protocols in place, the standard urine culture will not detect slow-growing organisms, fastidious and non-aerobic organisms, and most gram-positive organisms. The standard urine culture also can misclassify polymicrobial infections as sample contamination. It can take the standard urine culture up to 72 hours for a turnaround time. And last but certainly not least, previous exposure to antibiotics or the patient's urine exposure to antibiotics can skew the culture's results. And the bottom line is that just because pathogens have not grown in standard urine culture over the last 60 years does not mean that they were present in samples. And this has led to undiagnosed and misdiagnosed urinary tract infections, as well as antimicrobial resistance. So let's look at polymerase chain reaction and its advantages in urinary tract infection pathogen identification. The main take home that I want you to have from this presentation is that polymerase chain reaction is advantageous because pathogen detection and identification is not dependent on the organism's growth in culture. So let's take just a minute to look at a research study that compared PCR versus culture. In a retrospective record review of 582 consecutive elderly patients presenting with symptoms of lower urinary tract infection, all patients had traditional urine cultures, and they also had PCR molecular testing run in parallel. A uropathogen was detected by PCR in 56% of those patients. And by traditional urine culture, the uropathogen was only detected in 37% of patients. That's quite a significant difference when we're talking about clinical care of a patient. Now, the PCR and culture agreed in 74% of cases. Both 34% were positive and both 40% were negative. There was disagreement between PCR and culture, and that was 26%. The PCR was positive and the culture was negative in 22% of cases, and the PCR was negative and the culture was positive in 4% of cases. There are reasons why a culture may be positive versus the PCR being negative. That goes beyond the scope of this discussion, and we won't cover that today. We can cover that at a later time. In the 175 patients with polymicrobial infections, PCR detected 166 pathogens versus culture that detected 39 pathogens. So these results suggest that PCR may be likely to identify pathogens before traditional culture will. And this very last statistic is highly clinically significant in that polymicrobial infections were detected in 12% of the samples in which the culture had actually given a negative test result. So for 12% of those samples, or 12% of those patients, they may have been not treated at all for a urinary tract infection, when in fact, they had a polymicrobial infection. So let's just take a look at the clinical journey a patient would experience in which a prescriber, prescribes standard urine culture versus PCR for diagnostic testing. The standard UTI diagnostic workup looks something like this. The patient presents with signs and symptoms of lower urinary tract infection. The doctor performs a urinalysis via dipstick in the office. Based on the urinary analysis results, the doctor may send the sample out to the lab for a standard urine culture. The doctor will prescribe an empiric antibiotic before the patient leaves the office. And based on the standard urine culture results, the doctor will call the patient in 3 to 4 days to deprescribe, adjust, or maintain the empiric antibiotic. So the only change in this workup procedure when a doctor decides to use PCR testing is that they send the patient's urine sample for PCR testing at a lab that provides PCR testing versus sending for standard urine culture only. So you can see that PCR testing is not disruptive to the standard UTI diagnostic workup. Now, very importantly, let's look at the timeline for the patient, for the prescriber who uses PCR versus standard urine culture to diagnose their urinary tract infection. So let's say that Lab A has PCR, urinary tract infection testing, and Lab B has standard urine culture testing only. Okay, on day 0, the sample arrives at the lab, no matter which lab you send it to. On day 1 at the PCR lab, the PCR ID is reported, a preliminary AST is reported. However, on day 1 at Lab B, the sample is only being plated and to run for culture. What happens on day 2? At Lab A, the AST may be completed depending on the pathogen, but at Lab B, you can see that the sample is still running for culture. On day 3, at Lab A with the PCR testing, AST is likely completed depending on the pathogen. But down on day 3 at Lab B, the sample is still running for culture and the results might not be done. At Lab B, you can see that on day 4, the sample is still running for culture and the results might not be done. On day 5 at Lab B, the sample is running for culture, the results might not be done. And then on Lab B, the ID and the standard urine culture guided AST report may or may not be complete. So what is day 1? How does day 1 compare for the PCR lab versus the non-PCR lab? There's a faster turnaround time to accurate identification of pathogens or the absence of pathogens. There's also a faster turnaround time to deprescribe or prescribe. How about day 2 or 3 for the PCR lab? The PCR-guided AST has accurately validated or invalidated the prescriber's original treatment selection. So this leads to a faster turnaround time to prescribe or deprescribe. Let's look at the standard urine culture day 4, 5, and 6. A slower turnaround time to less accurate identification of pathogens or absence of pathogens. We also have a slower turnaround time to prescribe or deprescribe. So you can see from that timeline that the PCR-guided urinary tract infection testing produces a faster turnaround time for results, and it also leads to a faster deprescribe or prescribe. So let's just look at a summary of how PCR UTI testing can interrupt the progress or the typical scenario, which is where standard urine cultures that are undiagnosed or misdiagnosed can lead to the negative clinical and economic outcomes. If we use PCR instead of standard urine culture for the identification of uropathogens, we're able to decrease the negative clinical and economic consequences that we see from the standard urine culture. So now I'm going to hand over the presentation to Dr. Mayil, who will discuss with you the rigorous scientific and regulatory validation that PCR testing must undergo. And I want to highlight before he starts how critical it is to clinical outcomes from urinary tract infections that these tests undergo this scientific and regulatory validation process that he is going to describe for you. Okay, Dr. Mayil.
Thank you, Dr. Winslow. So any assay we develop, it has to go through 3 processes. First, it's going to be development, then assay validation, and then regulatory approval. These are 3 processes. So when you do the assay validation, we are establishing 3 facts. 1, the assay performance, we learn ourselves, and assay limitation. And third, of course, you have to get the regulatory approval to run the assay. So within that mind, in Acuities, we approach this problem entirely different. We look at the assay as an entirety rather than part by part. For example, in urinary tract infection, let us define first the matrix. Then we have to define how we collect the sample and transport. You have to produce a document, very clear document to regulatory agencies, how we are going to do it. Then the sample requisition form, how we are going to supply this to the clients. They want to see how it looks like. Then in the SOP, you have to have a clear definition for sample acceptance and rejection criteria. Then sample stability should be defined as well, which is we are going to validate when you do the analytical phase. Then based on the matrix, we have to define what are the interfering agents. Then extraction process, PCR, and results acceptance criteria. Then we have to submit a report. It's not just a single report, at least minimum 2 report, positive, negative, on top of that, based on our assay validation, any interfering agent, which is lower your LOD, or any cross-reactivity. You have to give additional example report, then clearly defined proficiency testing. Next slide, please. Next slide, please, Dr. Winslow. Thank you. Then we are preparing for the validation. When we prepare for the validation, we have to develop a validation plan. When you develop a validation plan, the first thing, of course, your testimonial. For that, you have to collect the literature. You have to do a literature survey to show the clinical relevance for the particular analyte. So you have to submit those literature to the regulatory agency. Then here, what we do normally, we do the theoretical LOD calculation. The reason is we want to take the assay performance in our hand rather than dictated by itself. At the same time, it also going to guide you if there is any problem in the validation, where to troubleshoot. That's where our success here. Next slide, please. When you prepare for the validation, the first thing, of course, matrix, the regulatory agency, they want to spike the material or use anything in the assay is a negative matrix. Here, the normal human pooled urine is commercially available. We also bought it for the commercially one, but we test it out on the culture plate as well as in the test system here in the UTA test menu. Make sure that everything is negative. Then we use the matrix for our experiment. So the regulatory agencies, they want to see the tighted intact organism. There is no synthetic DNA involved here. The only one case they are okay with that is a genomic DNA, for example, mycoplasma. It's very difficult to culture. At the time, they are okay with the genomic DNA. Other than that, they want to see all analytes in intact organism. And it is right now available from different sources. I listed few here. You can be able to get. Next slide, please. So the validation plan should include specificity, include like inclusivity, exclusivity as well, and checkerboard, sensitivity, precision, assay interference agents, accuracy, and stability. These things should be included in the validation plan. Next slide, please. So on top of this validation, the first thing we focus on the extraction. We all know that not all microorganisms are not going to lyse equally. For example, gram-positive organisms are difficult to lyse. So to overcome the issue, we are going to usually optimize the extraction. For example, if you have a drug-resistant organism, gram-positive drug-resistant organism, in some cases, the cell wall is modified. So those organisms are not going to be lysed by lysozyme. In that case, we may have to consider like mutonolysine, lysostaphin, depends upon the organism. So we want to optimize first to make sure that we have the ability to lyse and isolate the nucleic acids. At the same time, we also focus on volume of beads if you are using any bead-based extraction and the protease gate. Of course, we pay attention in elution volume, which is going to dictate finally for the sensitivity of the assay. Then we focus on controls. Apart from NTC and NAC, we pay attention in detail for the spike in control. The regulatory agencies don't like to see one CT value and below. They want to see what is our acceptable CT range. We have to run the experiment and show this is your acceptable CT range, which is also applicable to your positive control as well. They want to, you have to define very clearly in our validation document. Next slide, please. So we always run the specificity experiment first, especially the inclusivity experiment. If you are using OpenAI, it's an added advantage because it's a spatial multiplex. So if you are testing one organism, that means you are testing against 55 other different targets. So that's an added advantage here. On top of that, you have to select a few more organisms, genetically related organisms, commonly found in the matrix to demonstrate your assay performance is really good. Don't forget to include human DNA. The reason is there is not just a specificity, sometimes a human DNA can interfere in your assay. So one experiment can be reused in the assay interference agents. Next slide, please. So after you run the specificity experiment, then we move to the sensitivity. The sensitivity requirements are 6 to 10 point dilution series. Wait a minute. My test panel is too big. Usually UTI is going to be more than 30 organisms. That means there's a lot of testing. How we overcome this? So what we do is we make a pool of organisms. When we make the pool, we want to make sure that they're genetically distinct. Then you can run the sensitivity. This way, we can reduce the time and reduce the amount of money you spend to run this experiment. Usually, we combine LOD and precision together so that your validation progress really fast and save money. Next slide, please. So accuracy, there is a, do we need a clinical sample? No. Contrived sample is accepted only if you spike those material in negative matrix. It should be spiked in at least 2 different concentrations, near and above, one above the LOD. They need they want to see minimum 30 samples per analyte. So on top of that, you need a 30 negative sample for the 2x2 table analysis. We collected some reference material from another laboratory used in the experiment. When you do those things, collect the report as well. De-identified report you need to further summation. Next slide, please. Then stability. The stability must be defined in 2 ways, one at a room temperature and at refrigeration. Plus, you need to establish the nucleic acid stability. What we did here, we did 4 times freeze the cycle, then we analyzed, and then we demonstrated the nucleic acids were stable. When finally you do the data analysis, we have to pay attention in detail. It should be analyte specific. And you have to present every single data in a table format in clear way. With that, I will show you one last slide which contains some clinical data. Next slide, please. So these are not reference material. These are actual clinical isolates. These are, we grow it in the lab, and we took the live organism, we spiked in the urine, transferred to the collection device in 2 different concentrations, and incubated at an ambient temperature and in cold, in the refrigerated conditions. Then we lysed the cells, we ran the PCR assay. What is interesting I want to show you is at the higher concentration, over the period in ambient temperature, the CT value is going down. That's an indication of that this organism is growing. We all know that the urine is nutritious. They tend to grow. So why I'm showing this? The intact organism, live organism, when you include in the validation, you can understand the assay performance really well. Whether clinically this is going to affect the patient, the answer is no. The reason is most of the organisms are tend to grow when we spike in 100,000 CFE. But it will give you an idea what is going on in your assay performance. Plus, in using intact organism, you are defining not just the PCR, include the extraction as well. You are demonstrating you have the ability to lyse the organism and extract the nucleic acids. Thank you. Dr. Winslow, can you please take over?
Yes. So just to summarize my information and Dr. Mayil's information, I want to state that it is very important. UTI diagnostic methods absolutely matter in the clinic. So we have the patient, we have the healthcare provider. The healthcare provider, of course, is going to do a clinical evaluation. But the next step, choosing the right test for the right patient at the right time, what we know as diagnostic stewardship, is absolutely critical for the clinical outcomes that we will have on the other side of the diagnostic test. By choosing a rapid diagnostic test for urinary tract infection detection and having that rapid diagnostic result reported, as well as the right interpretation of the information, the right antimicrobial choices at the right time, will lead to those positive clinical outcomes that we want to see from the diagnostic method that we choose for urinary tract infection detection. So thank you for joining us today. We're going to now open the floor for questions.
Okay, thank you so much, Dr. Winslow and Dr. Shanmugam, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now by submitting it into the Ask a Question box. We'll answer as many of your questions as we have time for. Now let's get started. Our first questions here is, was nucleic acid extraction, was it done from the isolates or from urine specimens?
So we spike the organisms in the urine and we spun down, we collect the pellet, which can be used for extraction, not from the urine specimen directly, so that we can avoid any interference agents in the urine.
Thank you for that. Another question here. Clinically, we are taught to not treat polymicrobial culture or PCR reports. What is the clinical value of treating polymicrobial PCR reports?
Great question. So I think the point of that question that's the most important is treating test results. We don't treat test results. We want to treat the patient. So the patient should have clinical signs and symptoms of urinary tract infection for us to prescribe and use these test results. So we wouldn't encourage anyone to treat necessarily just test results. Clinically, we want to treat the patient and their clinical signs and symptoms. I couldn't hear Emily.
Oh, apologies. Sorry about that. Here, I'm going to repeat the question right now. Please discuss the denials by MolDX for PCR UTI panels. Palmetto and MolDX have not approved any UTI PCR panels for reimbursement to date, even those with a Z code.
I'll take that one, Dr. Mayil. In my discussions with MolDX about approval of technical analyses and reimbursement, this is coming from them. They don't feel that the literature that is available currently is sufficient to validate the clinical utility of the test. And they highly encourage the industry to join together to produce and publish the data that they want to see regarding clinical utility so that they can determine to reimburse the test.
Great, thank you. Right, another question here. Should the xenospike and control be added to each patient sample to ensure amplification? Should the extraction control organism be added to each patient sample to verify extraction?
I will take that question, Dr. Winslow. Yes, you have to spike the xeno control. In this case, we are using the live organism, the Bacillus atrophius. So it can serve both for the extraction control and the xeno control. So that's how the system works. So if you want to use any other controls, you can, but you have to do the extraction process. That's a requirement.
Thank you for that. Okay, another question here. SUC typically identifies organisms at less than 1,000 SFU as the lowest level of detection. With PCR, we can identify organisms down to 5 SP or 50 CP, apologies. What is significant in identifying the organisms to this LOD? And what should be the lowest LOD reported, or should we use as our cutoff?
I will take that question, Dr. Winslow.
Please.
So here in the UTI, yes, the PCR is too sensitive, of course, but if you use an open array, we call the sweet spot. The reason is when we ran the LOD, we came to the conclusion that it is not too sensitive. We have the ability to control the assay in such a way that we have the ability to produce 100,000 CFU or a little bit below. Not like 100 CFU per ml. No. Which matches with the CDC and the FDA recommendation CFU values. So it's not too sensitive assay. And we can have the ability to report clinically relevant organisms in a relevant concentration. Did I answer the question correctly? or you need more information on that?
We can let them respond if they have any further questions. Thank you. Here's another one. How does PCR ABR compare to traditional urine culture results for antibiotic resistance determination? And do you feel molecular ABR testing has the potential to replace urine culture in the long run?
Mayil, you want me to take that one?
Sure, please.
Okay. So antibiotic resistance testing gives us a genotype for that organism. So based on the genotype alone, we could possibly assume that the organism is resistant to that antibiotic. But what we also know is that there are many factors that play into whether a gene expresses itself phenotypically the same way. So antibiotic resistance gene sequencing, I don't feel that it will absolutely replace urine culture in the long run, because there's, like I said, so many other factors that go into whether that gene expresses itself. The other organisms in the microbiome that may be interacting with each other and such. So that's a short answer for that one.
Yes, I'd like to add a few more things, Dr. Winslow. So when we do the PCR testing, it's not going to be a real time, right? It's we are collecting the material, we are going after the genetic material, correct? So maybe the other organism may be dead, but still you may have the genetic material you are detecting. So at the time, you will not get the appropriate answer for the clinical treatment. It will always better to the culture and do the AST, and you can give the right information.
Thank you both. Some great questions coming in. Lots to get through. Okay, another one says, thank you, that was a great presentation. How far can genotypic AMR go in terms of treatment selection? In other words, does AMR resistance gene screening go far enough to inform a treatment plan?
Dr. Winslow, I will take this question.
Absolutely.
The reason is, yes, the reason of detecting the AMR, antibiotic antimicrobial resistance, is really good, so that you can avoid by some antibiotics in the early stage. The PCR results will come early, so that you can avoid, oh, I see the ACT or AMC, I can avoid these antibiotics. At the same time, the final result coming from the lab will support the antibiotic selection. So the AMR is not going to replace the routine testing, but at the same time, it will give you an idea which drug you should not use. Thank you.
Thank you. And just a reminder to the audience, if we do not get to your question, they will be answered by the speakers after the presentation. Another question here, how did you determine what targets are detected as pathogenic versus normal flora by PCR?
Mayil, you want to take that one?
So, absolutely, Dr. Winslow. Yes. So that's a very good, great question. Yes. So normally this question comes up when we are dealing with the coagulase negative, right? So that's the one is going to give you trouble. But when you have the assay, when you do the validation, you have to make sure that it reaches the clinically acceptable CFU value. If you do that, then you have the ability to overcome those issues. There are some other bacteria, Corynebacterium regali, that's a common human flora, which may give you trouble. But you are going to give you data which is going to substantiate that the CFE value is matches with the clinically relevance. Your assay cutoff value and how you run the validation is going to dictate those things. At the same time, of course, the clinician is going to look at the data and the patient condition before they start the treatment. Of course, it is up to the physician to decide. Thank you.
Great. Thank you. Next question. What do you think is the main hurdle for UTI to be a standard of care as opposed to SUC currently? And how do you see PCR getting there?
I'll take that one, Dr. Mayil.
Yeah, please.
Working on the clinical side of this test, in my personal opinion, and based on comments I've heard from payers and such, we have standard, or prescribers are taught to use the standard urine culture. That is the current guideline from IDSA and other professional bodies that provide guidelines on how to diagnose and how to treat urinary tract infection. In my opinion, I feel like as an industry, we are going to have to educate those guideline makers so that they can realize the value of PCR over standard urine culture. In my experience, prescribers they like to prescribe by guidelines. So if we can get the PCR into those guidelines and name where it's appropriate, how to use the test, how to read the test, etc., I feel like it's going to be about education and showing evidence to get PCR to be the standard of care.
Great question and answer. Thank you. Okay, have some few more questions. What type of tubes were used for the validation, and what is your opinion on preservative tubes for stability?
I will take that question, Dr. Winslow. Yes, we use the boric acid tube containing tubes for the sample collection and for the validation. The reason is our lab is a reference lab. The sample has to come from different places. So the stability is going to play a role, and we use the boric acid tubes. To answer the question. I hope so.
Another question someone's asking, in the validation discussion, I'm not familiar with checkerboard. What is it? It was stated to use 30 samples per analyzed. Does that mean 30 samples positive and negative, or poor organism?
Okay, I will take this question, Dr. Winslow. So you have two questions now. The first question I will answer, checkerboard. If you are using automated instruments for extraction process, the first thing it pops in mind whether sample is jumping from one well to another well. So what we do normally, for example, you are taking a 96 well plate for extraction. The well 1 has your sample. You usually spiked in 100,000, more than 100,000, or 10^6 CFU per ml, kind of high concentration. And the next one, you put just water, plain sterile water. So it's going to be zigzag. You put sample, control, no sample, just water, and then again sample, and then water. You do it like this, and then demonstrate there is no cross-contamination. This experiment we call checkerboard. Now for the second question, so you are talking about, I'm sorry, I missed the checkerboard. What is this? That is 30 samples per analyte. Yes, you need 30 sample per analyte, positive sample, which is 15 samples near the LOD, 15 samples should be above the LOD. That means, for example, 10,000 your LOD, 10,000 CFU per ml your LOD. Then you have to use 100,000 LOD, 100,000 CFU per ml is another 15 sample. And 30 urine sample you have to use, negative urine sample, so that you can do the 2x2 table analysis for the accuracy study. Thank you.
Thank you. Another slightly related question. Does the positive control have to be for all organisms on the panel?
Absolutely, yes. That's a requirement you have to you have to use. But once you show that even a synthetic DNA can be used for that purpose, it need not to be in a full organism. If a full organism is good, but you can use synthetic DNA, but you have to demonstrate, including extraction, how you are going to do the extraction and how you are doing the PCR. Then you are good to go.
Thank you. I believe we touched on this earlier, but someone's asking, did you use patient urine specimens for method validation?
Yes, we use for those things. Yes, of course.
Perfect. All right. Another few questions here. Someone is asking, can you really justify the cost of multiplex PCR versus culture for UTIs? It doesn't seem that the clinical value can justify the cost, especially considering that some labs are charging $1,000 plus for these tests.
I'll take that one, Dr. Mayil.
Yes, sure.
You know, this is how I like to answer this question because it does come up. I like to ask what is the cost of not doing the PCR test? What is the cost of false negatives? What is the cost of polymicrobial infections that are not identified? There are many costs that are associated with the standard urine culture besides the upfront cost of the test. So, you know, the question really goes back to, is the standard urine culture less expensive if we're not identifying or we are misdiagnosing urinary tract infections? I know it's not a financial figure, it's not digits, but that is how I like to answer that question. And that comes from the clinical side of this test. The clinician looking at whether they want to use the test, the standard urine culture versus PCR. What are the clinical outcomes, the negative clinical outcomes, and what do they cost associated with the standard urine culture?
Thank you, Dr. Winslow. And the next question, what is your opinion on spiking in the NTC control with extraction control organism?
I will take that, Dr. Winslow. The NTC should be just to plain sterile water. You should not do anything with that because that control is to monitor any contamination from your reagents. So you should not do anything with that. If you want to do the extraction control, then you have to introduce the NEC. That's why we have NEC, negative extraction control. There you can spike the spiking organism and extract and demonstrate. You can do the extraction. At the same time, the PCR is working as expected. Is it clear? Hope it is clear.
Yes. Another question, what collection devices, preservative media would you recommend for urine testing?
I will take that question. Dr. Winslow. So far, we are using the boric acid tubes. The reason is we are not only doing the PCR test, at the same time, we are doing the culture work, which we call PCR guided AST. So when you grow the organism in normal culture, we don't know what is going on. But since we are doing the PCR, we know which organism we have to look for. So we can leave the plate more than 48 hours and look for the organism. The reason is some organisms tend to grow slow. For that reason, we are using boric acid tubes. So it solves both the purposes. You can do PCR at the same time, you can do the culture work as well. Thank you.
Thank you. Next question is, how do you determine the cutoff value?
And the validation. Simple.
Perfect. Another question someone's asking is, how might we identify organisms outside of the PCR panel?
That's a great question.
Yes.
So most of the time we are seeing the PCR panel organisms. Very rarely, when you do the culture, when you do the culture, we are noticing some organisms. Of course, recently we encountered some Acinetobacter complex organism, which is very difficult to identify, by the way. And initially we missed in the PCR, of course. But since we are doing the culture work, we are giving the necessary information to the physician. That's how we are managing with that. So we are not going to miss the organism which is going to grow on the culture. At the same time, we are giving the results much faster than the culture work using the PCR.
Thank you. So we have time for a few more questions. Another one we have here is, would PCR be a good choice for patients with recurrent clinical signs and symptoms of UTIs whose standard urine shows no growth?
I'll take that one, Dr. Mayil.
Sure, please.
My answer is absolutely. What we're finding is that quite often patients that have clinical signs and symptoms of urinary tract infection, but their standard urine culture is negative, that the PCR does identify an organism that may be the causative agent of their clinical signs and symptoms. Yes, the short answer is yes.
Perfect, thank you. A next question is, I feel like the PCR test is too sensitive. PCR identifies microorganisms the medical community hasn't typically seen reported from standard urine cultures, and I am concerned about overprescribing antibiotics when they aren't necessary. What are your thoughts on PCR being too sensitive?
I will take that question, Dr. Winslow. The question is great, and I agree with them. PCR is too sensitive. Yes, but the sensitivity is going to be determined by what kind of platform you choose, how much the elution volume goes into the PCR reaction. These two things is going to dictate. In the case of urinary tract infection testing, we prefer open array. The reason is the limitations over there, which we can use as a system to do the urinary tract infection, which is going to be from our validation from between 10,000 to 100,000. So it's not too sensitive, and we are not violating any already established concepts.
Yeah, I'd like to add on to what Dr. Mayil said. As a clinician, my concern would be that I am under-treating, especially those patients that have false negatives but have signs and symptoms of urinary tract infection. Those patients often feel unheard. They are often confused by the fact that they know their body, they feel they have the signs and symptoms, but the tests are negative. The standard urine culture is negative. So that's my concern, or that's what I hear clinicians saying, is that, oh my gosh, if I think about how many patients had a negative standard urine culture, but they continue to have signs and symptoms, that bothers me personally. So that's the kind of feedback that I'm hearing from clinicians is even the fact that they may have been under prescribing for those patients.
Thank you. Another question here is, normally I empirically prescribe an antibiotic until the patient's standard urine culture returns, and then based on the AST, I know whether to change the antibiotic or continue the antibiotic. If PCR test results are available in 24 hours or less, should I refrain from prescribing empirically until after the PCR results are returned?
Yeah, that's $1 million question. But what I'll say is that the great part about PCR being returned so quickly, such a fast turnaround time, is that if the physician or prescriber wants to wait to prescribe until they get the PCR results, there's a very short turnaround time. If the prescriber wants to prescribe an antibiotic in the physician's office before that patient leaves, before any test results have come back, heck, before any of them have been sent out, they're also able to de-prescribe at that short turnaround time that comes from PCR testing. So, let's just say that a prescriber prescribed Septra for a UTI, and then when those PCR test results come back, there's no organism. This is hypothetical. They can de-prescribe that antibiotic quickly. So it's really, the physicians or prescribers, what works best for their patient population.
Thank you, Dr. Winslow. I'm going to end with this question. What does the payer landscape look like for PCR UTI testing? And do payers reimburse the test, or will my patients have to pay out of pocket?
So I'll take that one. The payer landscape is, of course, it varies based on the geographic location of the lab, for one. There's so many factors that go into the payer discussion and whether payers are paying that go well beyond this conversation. So the shortest, easiest answer is that it's highly lab dependent, your technical analysis, et cetera. So I think that's just the best place to leave that question.
Thank you. And thank you again, both Dr. Winslow and Dr. Shanmugam, for your time today and your important research. And do you have any final comments for our audience?
I just want to thank everybody for joining us. I think we had a great turnout, and I really hope this information gets pushed out to others as a result. How about you, Dr. Mayil?
Yes, the questions were really nice.
Yes.
I did not expect like this. It was real interactive questions. Thank you so much. Thanks a lot. That's really encouraging.
Yes, very interactive. Thank you both. We'd also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's webcast. And before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we were unable to answer today and those that come in during the on-demand period will be answered via the email address you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with any of your colleagues who may have missed today's live event. Thank you. Until next time, goodbye.